Hello, everybody. So my name is Thomas Larsson. I'm a bioinstrumentation at the MBIS, and my task here today is to talk a little bit about the QC and assembly, and a little bit also about the annotation when you set out to do a new reference genome. So my background is as a molecular biologist, evolutionary biologist, and then been working with assembly and annotation project for 15 years, I think. Yeah. So a little bit the scope of this talk is just to give you a little bit of an overview of the process from the raw data that we get from someone like Olga et al. And onwards to the assembly and annotation. A little bit of the kind of problems you might encounter when you try to do this, and also links to tools and some recommendations, and also where to get in contact with someone like me or my colleagues if you need some help or advice during the planning or later on during the process. And I also try, when it comes to the links and to the tools and the recommendation, you will see in the end, I was a bit afraid because this field is moving quite quickly. It's hard to give hard recommendations to specific tools I discussed during the lunch break. So my aim here for the links that I provide in the end are links that are likely to be updated in the foreseeable future. So, because chances are otherwise that when we turn off the recording, what I said is not taken risk. So hopefully it will stay relevant a little longer then. So then the first thing I think that is interesting to have clear in your mind when you set out to do this project, and Olga already touched on it, like maybe you need to know what you're aiming for. And Earth Biogenome Project standards are quite high set compared to many other projects that are traditionally performed by smaller research groups. And basically, these are the latest recommendations. So having contigs that are more than 1 megabase, essentially sequences so that half of your assembly are made up of sequences that are 1 meg or longer, an error rate that is 1/10,000, and then they have some additional criteria that you can apply also, like you should be able to map more than 90% of transcriptome data back, BUSCO completeness, I come back to that, but essentially what that is saying is, can you find the genes in this genome that you expect to be there? And also false duplication, that you remove haplotypes that should not be in the assembly, and also K-mer completeness, so you have the data that went into the project are actually present in the assembly, you see. And then ideally also you want to be able to name a scaffold that's come out and assign chromosomal name to them, so you know that you have at least 90% of your assembly assigned to specific chromosomes. With that said, for many projects, these are quite highly set goals, I would say, but it's good to aim here to begin with. But depending on, I assume there's a lot of different favorite organisms in this group of scientists here. So depending on your species and the conditions that you work with, maybe this may not be applicable with the current technology, especially if you work with samples that are tiny or where it's very hard to get enough material, for example. That said, I think you should still give it a go. That would be my take home. But before you start, and this is a little bit of a education, but I think it's important to resort the growth database, and a little bit of Googling in other places as well will save a lot of time. Is anyone else already doing something similar? So just to find out if there are other initiatives, and I think as Olga mentioned, or the first thing this morning, maybe you have to swallow your fear a little bit of being scooped and try to collaborate instead of making that two separate research groups make two so-so assemblies. Instead, you join your forces and make one really good one instead. That would be my goal, I think. And also with relation to the quality, I think if you cannot really meet the goals, that the high set goals of the CBP standards, maybe at least it's a good idea to have clear in your mind what are the main questions that I want to address with my reference assembly, because then you can essentially still, it will be easier to know when you're finished, it will be easier to know how big budget you need, and it will be also a little bit, how should I say, it will be easier to plan essentially. And there is a lot of things you can do with reference assembly that does not fulfill all those points, I should say. Especially if we work with a new model where nothing is known before. So that would be another thing. And then, of course, the more you know about the biology, the genome size might already be known beforehand. You may find old papers about the karyotype, and if it's polyploid or repeat content, whatnot. All that kind of biological information is nice to have, to be able to plan and to be able to spot problematic things along the way. Even like an old-fashioned genetic linkage map might help you, because it's an independent set of information that you can use. Yes, so we heard a lot about long reads, and I just want to introduce the problem of genome assembly in general terms first, because we talk a lot about long reads, but essentially what we're doing, even with the really, really long reads that we get today, like the bulk of the sequencing from HiFi, for example, they have 20 kb long reads, they are still just a minute fraction of the actual molecule that we want to look at. So they are long, but they are still short. And that causes some problems. And I take it to rather old references here, but they still illustrate the point. So what you can see the pointer here. So essentially what this shows is some sort of, here you have on top the genome, very simplified, and here is a graph representation of that said genome. And that is how most assembly software work. They try to represent the genomic sequence with some sort of graph, and then they traverse the graph in just one correct way, ultimately. That's what you're aiming for. And here, in this particular case, is those arrows here represent repetitive sequences. The two black ones represent the same sequence, and the gray ones represent the same, and so on and so forth. And then you end up with this representation. And the take home here is that if the reads you're using are shorter than these repeat units, you need additional information. And Olga mentioned MAPER libraries that produced a long time ago, but nowadays we have other technologies for breaching those gaps. And here is also just an illustration of the complexity of the graph representation given increased read length from 100 to 5,000 base. So you simplify the problem by getting more reads of good length. And that's, of course, a given, but I thought I'd mention it. So this is some sort of general overview. So when you get your data, you will do some sort of initial QC, and if you get the data from Olga and others, you will have a lot of that initial information, how many reads, how much data that is useful. That will basically be in the reports that you get from them already. But you may want to do some additional cleaning and looking for adapters and technology-specific sequences that you want to clean out first before you do anything else. And then you essentially go to config assembly, remove haplotypes, maybe look for contaminants, and also removing organelles. And then once you're done with that, you can go on to scaffolding of the assembly. And then depending on the technology you have been using, you may also want to do some polishing. And then again, once that is done, you can probably identify if you have contamination. Some of it might be easier to identify when you have assembled stuff with the longer sequences. And then you will do some sort of curation of this scaffolded assembly and remap the data, the high-C data in this case. And then there are some additional looking that you only have a single representation of each part of the genome, looking at the base accuracy, and also looking at BUSCO, which is a common tool for looking at the completeness, and depending on different taxonomical levels. Once you're done with that and you're happy with the result, then you can go on with annotation. And this, I will just skim over it, but I think general recommendations, these are essentially from Biogenome Project's recommendation now. So at least 15x coverage for each haplotype for contiguous sampling if you use PacBiodata, more if you use Oxford Nanopore, but once you're ready to run your project, check again, because it might change very quickly. And then more, and HiC data for scaffolding. These are the sort of general, but I will give you some links to more recommendations. And I think it's important to try to get, if it's possible, if you work in mixed species, where you can get all the data from a single individual. That is what you should aim for. And as we heard before, choose an individual from the heterogrammatic sex if that's relevant for the project you're working with. And then RNAs that are written from the same individual if you can, or at least from the same strain or as close as possible. And this is always the best evidence data you have for making good annotation later on. So if your plan is to annotate this genome, aim for RNA-seq, preferably from many, many different tissues and stages, and especially the tissues and stages that you're specifically interested in as well for follow-up studies. And I would suggest, as Olga said, also that it's not so expensive anymore to sequence. So aim at the higher so you can afford to filter a little bit if needed later on. But I think, yeah. So there are many different things that you can do for quality check. And a lot of, especially if you start out working with the species where little is known beforehand, you may want to and get some first peek on what this species might look like already from the raw data. And a lot of that type of analysis is based on k-mer counting. So you're essentially sort of decomposing the sequence into shorter sequences that also are shifted one base. I'm sure many of you are familiar with the concept of k-mer. And then you can get some initial estimate of what it looks like. For example, a tool called GenomeScope will tell you how large it might be, and if it's a diploid or a triploid, as you see in this case, and then how much is, as you can see on the sides here, if you have a huge part of the curve on the left, that would indicate that your sequencing library might have contain a lot of sequencing error. For this, you can already have an idea of the data before you start doing anything else. So I put the reference here. It also describes another tool that is called smudge plot, or a concept of smudge plots that you can actually use to guess or try to identify if there is a higher ploidy level. This might also be very useful initially in a project if you really don't know, especially if you work with plants, and they can quickly be polyploid. So you might not know that on the onset. Yes. And there are other things. This is from another tool that we are sometimes using, k-mer analysis toolkit, link here. Essentially what you can do there is compare the first slide here. It's also based on k-mer counting, and you can compare multiple libraries in your project to see if they are likely to come from the same sample, essentially. That will indicate if something went wrong with one or the other library. This could help you in the initial testing. And then also you can plot the k-mer content versus the GC content to see if it's likely that this is from one species, or in the case on the last plot here, where it actually seems like you have contaminants, because there are different coverage levels and different GC contexts. So a plot like this would indicate that your data is contaminated, or that you're working with the meta-genome. So those kind of things you can already get an idea about before they even start assembling. And then I won't talk so much about the actual assembly process, because once you have the raw data and you believe that it's in good quality, you throw it at one of the commonly used assemblers, for example, Hi-Fi SM, which is the go-to assembler that we use most of the time now for packed biodata. And after that, you get the set of pultigs and can run it through a tool like purge dupes, for example. And essentially what you want to get rid of there is duplicated, you only want to keep one haplotype. And based on mapping back the original data, you can get some sort of cutoffs where most of the data that is useful in your case is in this part of the curve, essentially. And you will get rid of the poor quality data. I will also get rid of repeats and junk sequences. I will give you a report, essentially, with sequences that are likely to be haplotypes or repeats or junk. And once you have cleaned it up, you can also screen for contaminants. These are two of the tools. There are other ones. Some of you might also be familiar with tools like Kraken, for example, that is commonly used as well. But essentially what you do, you try to match your sequences against the database, and you will get a taxonomic report of what is in that sample. And you can also do, in the case of blob tools, you can also actually get a graphical representation of things that might belong to different species based on camera content and coverage and so on, and GC content. So this is really handy, especially we saw this example earlier today when you sample a sponge that are actually two sponges. This will appear in such a plot. Yep. And then, as I said, with this document shredder image, what we try to do is essentially putting together a very complex puzzle with very small pieces. And then we have new technology now, or relatively new technology. So the scaffolding with high-C data is really making it possible to put together complete chromosomes nowadays. So this is from the paper that is a few years old now, but basically the underlying principles on why this work is that you get higher interactions within chromosomes than between chromosomes, and it's much higher signal close by in the chromosome, and you also have some sort of smooth signal, or at least that's the assumption. And then when you map these data from a high-C library and visualize the contacts, essentially which part of each genomes are located close to each other in three-dimensional space, you ideally end up with something like this, where all the signal is close diagonal. And this is a signal of a good high-C experiment. But in many cases, you get things that look like this. And you can immediately see from these plots that the contacts are a bit off diagonal, but by flipping the sequence, you can sort of smoothen this. And I'll come back to that in the next slide. Of course, this is based on the assumption that the high-C sequencing worked. And Olga showed one thing I have not on my slide here is if you get a lot of trans interactions, that might be an indication that something went wrong with the library. But you can also use different tools to parse the important ligation pairs. I won't go too much into the details. I put the link here to one of the tools that we are commonly using, called pair tools. I think it's that page has a lot of good descriptions on which part of the high-C data is actually useful for scaffolding. But what you aim for is uniquely mapping pairs, indicated up here. And then you have all kinds of cases where you lose some part of the data, where you have multi-mappers or non-mappers, and then we cannot really say so much as we can for the unique mapping pairs. And this is also how problematic this is is also dependent on the genome in question. So if you have a highly repetitive genome, you will get many more multimappers, and then maybe we need to generate a lot of more data to get the signal that you're aiming for. There are tools for visualizing the output from pair tools, and then you can sort of, what you see here, each line represents one library, and then you can compare the quality of the different libraries. And also down here, the same libraries, looking how many that has cis pairs within certain range. There, of course, what you really want is the more long-range information for the scaffolding. They will end up with something like this. So on the left side, this is from one of the tools we use for editing high-C scaffolding data. This is called user tools or usebox. I will show other ones in the end of the talk. But essentially what you see here, if you just flip this square in the middle, then you will end up with this contacts close to here and these contacts close to here. So, So this is an indication of where you actually scaffolded a piece in the wrong orientation in the assembly. That's better. And sometimes it looks much more complicated depending on what you have. And for example, here, these kind of off-diagonal lines are a classic example of where you haven't removed the haplotick. So this is so there is a bit still on this type of the analysis. There is some hands-on work, and this is what it might look like in a reasonable case. So here you see the whole map where I actually managed to get more or less all the chromosomes here. So on the left side you have the map that comes directly out of my scaffolding tool, which is JAWS, yet another scaffolder, high-C scaffolder it's called. But you can see there are some parts here that does not seem, so each square represents essentially one full chromosome. And then there are some shrapnel in the end, which is not assigned to any chromosome. But just with a little bit of editing, moving things here to the chromosomes where they have the most intense contact, you see I have just a tiny fraction that is not placed on any chromosome. This is sort of manual work, but it's pretty, compared to using mate pairs, what we could 5-10 years ago or something like this, this is science fiction, I think, but running through and get the chromosomes directly. Yeah. Okay, and then you can use, again, once you have that, you can go back. This is a tool called Mercury. It can do a lot of different things, but what you can do is actually compare in your final assembly the k-mer content of the read set, and then you can compare the k-mer content in the assembly or your different haplotype assemblies. And it can give you a lot of information on how well the assembly you have is coming from the data used, how well it was working. And probably there are many more examples here. I won't have time to go into all of that. But one thing that is really important from this tool is that you can also tell you k-mer is found in the assembly but never present in your in-data, meaning misassemblies. That's a good indicator of quality. Yeah, and then more of a biological test of quality is the BUSCOS, which is basically looking at one-to-one orthologs in the different assemblies. And here is one example where I have looked at the metazoan level. You have different taxonomical levels where you can score the assemblies, and basically telling me if it's likely to be complete, if it contains a lot of duplications or fragmentations. And I think the most important part in here is not to blindly look at the actual numbers. I think you can use it for evaluating multiple assemblies in the same species from the same data. It doesn't matter so much if it's 92% in your species and 94% in someone else's species. It's more important a tool to choose which of your own assemblies are the best, I think. Yeah, now just briefly something with regard to annotation. There are several different tools. So in order to annotate your genomes once you got to this point, what you need is some sort of information about repeat content. This you can generate with tools directly from your assembled genome. And then you usually use transcriptome evidence and also from related species and reference proteins. And you can also infer genes just based on the sequence content, try to what is called ab initio gene prediction. And there are tools such as Maker, for example, that will just try to merge all this information into one set of good quality gene models. And then once you have those, and maybe I should say, so here is where the multiple stages, the multiple tissues of your RNA comes into play. And then, of course, you want to assign functions to them. And chances are, of course, that if you have a genome that you deposit to a public repository, they will probably annotate it once again for you when you submit it, but that could take some time. So you may want to have some preliminary information at least. And the minimal thing you need is the gene models from a structural annotation and a reference database of known proteins. I have put here two tools that we routinely use for making this functional annotation. It's called IntProScan and Edinome Mapper. They're pretty straightforward to run. I'm sure some of you are already familiar with this. Yeah, so in the end of this process, hopefully then you will have one resulting assembly in FASTA format that you're happy with. If you're able to choose one of all the multitudes of variants you might have arrived at. And then you will also have a resulting annotation in GFF format. These are sort of the minimal output, but there are other things as well. And Stefan will talk about how to deposit these files into public repositories in addition to their actual raw data. And I think Daniel will also tell us how we can use those files to visualize them in the genome browser in order to get an idea of what the genome looks like and share it with collaborators and so on. So I will try, as I said in the beginning, try to put some links that are not outdated as we leave the rooms. So I start with our own in-house, and here I want to give credit to the right person. So Mahesh is the one that has been driving the development of this assembly pipeline, and with help from Martin and many others in the team, especially Martin worked a lot on the high-seq part of this. And so this is a good place if you want to look what kind of tools we are trying out. This is still work in progress, but you will at least see which tools are we routinely trying and using for our own in-house projects. And then I also put links to the Earth Biogenome project, like what kind of assembly standards they recommend, and that will also continuously be updated, and also report on assembly recommendation. There we will find a long list of tools that they recommend, also depending on what kind of data type, if you use hi-fi reads or if you use Oxford Nanopore reads, or what kind of technology. And this is constantly updated, so I think that's a good place to start. And also some links to Sanger for some of their pipelines. So, and also for the high-seq part, I put the most common tools that we use to visualize high-seq data or to edit the high-seq net. They are all pretty nice. PTextView is the one that we currently use the most. And then, again, if you want to look more into the annotation part of things, these are the, I think, very good places to start. And also, we have a few different tools. And I think even if Maker is a tool that we've been using quite a lot, but has maybe fallen out of favor compared to Breaker. But I would still recommend you to read, if you're new to this field, a lot of the information on the Maker homepage or the information on different Maker workshops is really, really important and nice to get an overview of this field. So I can recommend that. And then when you run into problems or when you want to discuss how to plan a project, contact us at MBIS. And we have one thing which is a sort of least level of resistance in order to get some help is to join our drop bills every Tuesday online on Zoom. You will find it on the homepage here. And then basically you will get together with one of our experts and they will guide you. And you can also get a couple of hours free consultation to discuss a project before you start. So this is free of charge. And I put some other links that might be useful to some of you. And also, if you're a PhD student, have a look at the advisory program. If you have a project that has a lot of bioinformatics work to it, then you may want to join our advisory program, but you will get one person from NDIS as an extra bioinformatics advisor. That might be a good way to come into this field if you are from a different field originally. I think that pretty much on what I wanted to say, and thank you so much for listening.